Okay, the unit outline's on the web, so I haven't printed one out for you just to save paper. So I'll go through the key parts of it now. The first thing is I've tried really hard to have a correction to the volume of the recording, so hopefully the recording volume will be better this semester. I've asked for advice as opposed to just doing it myself without knowing what I'm doing. So can you check today? If it's no good, let me know. If it's good, let me know and say thank you, be good. So the whole unit this semester is really into three little bits. We'll do one section of mycology, a small bit of virology, and then finish up with some parasitology. So we have two practical exams, one for mycology and one for parasitology. You must pass both practical exams to pass the unit, and you must pass the final exam to pass the unit. So we'll have a couple of small quizzes, not worth much, mid-semester test, a couple of assignments, and two separate practical exams. And we'll hold this exam for mycology during the semester, so it's still fresh in your mind, as opposed to doing it at the very end of the semester. So there'll be two smaller exams, not one big practical exam. When it comes to the assignments, if you're doing the Masters or the Grad Dip, then these quizzes are worth nothing, and the assignment's worth double the amount, worth 20%. Slightly longer assignment, a bit more involved than for the undergrads. The handing dates there are reasonably fixed to try to make sure that we get things in on time. This one's important because when we come to do our student presentations where you come and present your stuff to the class, I need these at a certain time to make sure I get all the information together and can plan who's going to present on what day. So this day is pretty much fixed. Everything though is flexible if you ask me nicely enough. They're all self-explanatory. That's all good, that's all good. So the key things about passing the unit, you must get over 50% for the whole unit when I add all the marks up together and divide them by what they're worth in weighting sense. But you must pass the practical exams, and even if you get a great mark for everything else, if you fail the practical exams, you either come back and do it again until you pass it, or you can fail. So it depends on how bad you got on the practical exam. Remember, the subject really is a practical subject, and the profession is really practical, so if you can't do that stuff, I don't care how smart you are on paper, you're not going to pass the unit. You must submit everything during the semester. If you don't, I might make you ineligible for the exams. And the pracs are compulsory. You can't just come and go when you feel like it with the pracs. The major reason why you fail the prac exam is you don't turn up. But they are compulsory, and if you decide not to come, I'll be keeping a record of who comes and who doesn't come. If you aren't there for pracs for any reason, without letting me know, then I can make you ineligible for the exam as well. Even though I keep saying that you can put the assignments by Milo Dropbox, don't. Just email it to me. I can't do Milo, I don't like it, I hate it. So just easy with the email. One day I'll get around to using Milo, but for the moment just email it to me. What's that? Yeah, we should, I agree. Referencing, for all of my assignments, reference it to the journal called the Journal of Antimicrobial Chemotherapy. Their style is a style I use for everything that I do, so just use that one single style, become used to it, and it's just easy for you, as opposed to saying, this time do this one, this time do something else. Just do one style for all your referencing, should be fine. Go past all of that, go past all of that. Yeah, go past all that. Textbooks, there's not really a good textbook for this unit because it's so disjointed in the sense that we've got these three small modules. The book you had last year or last semester for me, Michael Away, will cover some things reasonably well. Things that aren't covered in your textbook, I'll give you handouts for them. So it should be fine without a textbook specifically for this particular unit. That's all okay. That's all rubbish. So the timetabling for the unit is really annoying. I hate the idea that we have this 2-hour lecture and then straight away a 1-hour tutorial. I've tried for years to change it, we can't change it, so we're stuck with it. Doesn't work particularly well. So most times what I'll try and do is I'll do a 2-hour lecture, and sometimes we'll have a bit of a break and then come back and do another 1-hour session. Sometimes in shoot time I do it to do corrective material, other times we'll just do a bit of a chat, and sometimes we'll just go right through and finish early, say 2 hours, 20 minutes. I try and avoid doing 3-hour blocks, it's just too long for me and for you. You'll go to sleep and get sick of it. So we'll try and be flexible, but most times we'll be finishing well before 12 o'clock. Occasionally we'll have a decent break in between, so quarter of an hour or so, and then come back and do a third hour as a totally separate thing. The recording will be in 1 3-hour block. So if you do have a bit of a break, I'll just pause the recording and then come back and restart it. It's all good. It's all good. It's all good. Okay, the timetable, the schedule. So no pracs this week. Yeah, because that's already got no yesterday and the day before. So we can't have pracs this week. Then we'll have 5 weeks of pracs for the mycology, followed by the prac exam for mycology. We'll have a mid-semester test, and then we'll go over the test straight away, and I'll try and mark that quickly for you. After the break is when you'll be doing the student presentations. So one of your assignments is to do a case study. I'll talk about that in a minute. But the students present their work in these times. So the prac times for this week and that week are not pracs. They'll be in here, and you'll be doing a student presentation. So even though the prac times is a 3-hour slot, on Monday, rather than being 2 to 5, it'll be 2 to 4, because doing 3 hours of student presentations is just too long, because often you're not very good and it gets a bit fast after all. So we'll do 2 to 4, and then Tuesday 10 to 12. Where possible, it's best if you come to all the sessions. I can't force you to, but if you aren't here, so if you are in a Monday prac group and you decide not to come, that to me is being absent from the prac. So for the session you must attend is your prac group session. I encourage you to come to all of them. You catch up in the end with free time anyway. So there'll be those sessions there. These lecture time here and this tute time will also be student presentations, and then we'll go back in a bit more virology. Then the last part of the semester is all about parasitology. This to me is the best part of the unit. I really enjoy parasitology. It's mainly microscopy, but it's quite interesting. This week 10 slot in the prac, there will be no prac here, but the Monday is a lecture in the prac time in this room. The reason that it's got to be in there, it doesn't fit anywhere else. I want to try and make these things work in a sequence to fit for the pracs that are coming up. The parasitology prac exam, not sure when we'll do it yet. Come near the end of the semester, it's possible we can do this in SWAT back and get it out of the way, or we'll put it in the exam period. Once it goes in the exam period, I lose control of what we do because it goes to the exams office, and then they put it where they want to put it. If we do it in SWAT back, it's up to us to decide how we do it and when we do it. So most years, students prefer it in SWAT back and get out of the way, and it's just one less thing to do in the exam period. So nearer the time, we'll discuss that and make a decision together about where to put that exam. It's probably only about a 2 or 3 hour exam. It's not a huge thing. It's only on one day as well, so it's not such a drama. Also, if you do it in SWAT back and somebody fails it, there's time for you to do it again and still pass the unit and graduate and all that sort of stuff. If we do it in the exam period, it's done formally. Then I've got to wait for the exams office to put the results out. It takes weeks and weeks to do that, and then your sup or your deferred, whatever else comes much later on. So I prefer that exam to be in SWAT back because it's just our control of it then. I think that's all pretty much okay. Was it all good? No questions, comments? No? Right. So, the first assignment on mycology. I think about assignments sometimes. I think we often make assignments just to have something to do. We go, oh, I think an assistant Python, give them an assignment. It doesn't mean much to you in terms of ongoing usefulness. So this assignment is meant to be something that will really help you learn the material that you need to pass for the exam. So this assignment is about you writing a mini laboratory manual on how to do a key part of mycology. So in mycology and routine labs, the most important thing that you do, the most routine thing you do, is look for tinea. So this assignment is writing a method manual that helps you detect and identify the fungi that cause tinea. And you're writing it in a way that I could read it as a novice and understand what to do. So some of the key things on here is up here. Sometimes the students go, oh, writing a manual. Is it, do I tell you how to do something or tell you what to do? And what to me is important is you understand that this assignment is about you telling me what to do, not how to do it. So, as an example up there, I've said, it may be that you do a test called a KOH digestion test, where you put a bit of toenail or fingernail in a glass tube and add some KOH. What I don't want you to do is tell me how to do that test. Don't go and then put the toenail in the glass tube, then add some KOH and incubate for an hour. That's a separate method that in your lab would be the method manual. This is an overall scheme that tells you how to process a specimen. So you might say, for all pieces of nail, either toe or finger, please do a KOH digest. So that is telling me what to do, not how to do it. So it's reasonable to assume that from this manual here, in a real lab you'd refer to other sections of the manual that have specific methodologies about what to do, about how to insert in your test. So to make this more about what you do in what sequence to achieve the outcome, not how you do each individual little step. There's some leeway here. It's not a set way of doing a manual, but I want the manual to be written in a way that you assume the person has some knowledge, not off the street, they've got training, but I'm not sure what to do. So right now, if I went back to a laboratory, I would know how to do most things, but I wouldn't know how you want me to do in your lab. So do you want me to do this, this and this, all these specimens, or this, this and this, and all these specimens? So that's important you try and get this right. There's some terms of reference there, so make this lab manual cover these organisms, and only these organisms really. These are the ones that are most commonly seen, and to make your manual cover more than those is making your life too difficult for no reason. Do the common things well, and your life is really easy. Don't spend lots of time fussing about things that are so uncommon. When it comes to the guidelines, it just makes my life easier if you do it the way I've asked you to do it. So if you don't, then you lose marks for annoying me. So try and make the font the right font and use the right kind of setting out. The line spacing just helps me have everything in one way to mark them. And you must have these sections in your assignment. So there'll be one called background, one called requirements, one called specimen collection, direct microscopy, culture, ID. And you must have those in there in that way. It must be 5 pages, and the reference page isn't part of those 5 pages, but it must be on a separate page. With the background part of the assignment, all this will be is a paragraph or 2 maximum. When you come to a manual and just go straight into what to do with no context for why you're doing it, it's hard to understand. So if you were doing a manual on how to do a bilirubin test, you'd have a bit of a spiel about why it's important. Same here. You might just do a little spiel saying that these organisms cause tinea, it might involve this tissue or that tissue, and that culture and identification will guide therapy. Simple as that. Just a little simple statement that puts what you're doing in the context. For the requirements, again, some leeway here. Requirements to me is nothing is worse than sitting down with a manual of what to do, getting about 3 quarters away from the manual and going, oh shit, I need this, this and this, I haven't got them. They're down there somewhere else or they're in storage. Requirements tells me what things I need to have on hand to do all this stuff. It's reasonable to assume that some things are so commonly available you wouldn't put them in there. So it's a bit dumb to say glass slides, because every lab has glass slides and they're always going to be everywhere. But you might want to say a fluorescent microscope if you want to use one of those. They wouldn't always be everywhere, would they? So some things, again, will be grey area. Don't overdo these requirements, but you say cover slips and glass slides, because everyone's got them anyway. Think about what you want in a manual. If you're trying to do a test in your prac, what you want are simple, really clear instructions, often flow diagrams or charts. You don't want reams and reams of text because you never read it. Friday week 6 is preferable, but sometimes I understand you might get a bit stuck with other stuff, so I can actually accept week 7. Just depends. Really important for me is when you hand these things in, that the document name you give me has your surname and then underscore mycology assignment. You might think this is funny, but for me, getting 30 assignments in by email, when 7 or 8 of them come to me with assignment one with no name, does my head in. And often I've got to go back into the document, open it up, go to options, find preferences, and see who owns a bit of software, because there's no name anywhere on the whole document. That wastes me quarter of an hour. So put your name on it, makes it a lot easier. And there's a rough guide as to how I'll mark it. So I want to look at the content. I'll make sure you've covered all the pathogens, good references that are reasonably up to date, and you've done things and you comply with instructions. It's so easy to lose marks by not doing what I asked you to do. And that is just so easy to fix up. Just read the assignment carefully and do it as I asked you to do it. It's easy. This is pretty important though. You can have a document that's got every bit of detail under the sun, but it's hard to use as a manual and it won't get good marks. It's got to be easy to use. Now I understand that what I find easy to use and you find easy to use might be the same thing. I'll try and be fairly careful, but in the end it will be quite subjective. The other assignment is the case study. And for this case study, I want you to go to literature and find a case that you find interesting. It need not be mycology. In fact, it most likely won't be mycology. It can be parasitology, it can be virology, bacteriology, or mycology. It doesn't matter what it is, as long as it's a microbiology case. You may find just one single case, one person with one particular infection that you find interesting for a particular reason. Or it may be a case series. There may be 4 or 5 cases that are linked somehow. Or it might be a whole outbreak, a big outbreak of a disease in a certain context. In the end, as long as you found it that's been published, then if it's publishable, then by definition it's probably interesting. You can't go and publish E. coli causes cystitis because no one wants to know, because we all know it happens. So you can find these things for yourself. Early in the semester, come and see me and or email me and let me know what you're thinking of doing, so I can go and troubleshoot and quality control and make sure your case is A, interesting and doable. Sometimes we'll choose things that are so esoteric, it's hard to make any sense of them. In the case study, you'll do a PowerPoint presentation and you'll present the case plus some background information. It can't just be you find one article on one case and that's it. It'll be one article on one case, which is your basic article, that you'll then find other articles to support that to give you some background information. So it can't just be one single reference, it'll be more than one. If I find a case of scedosporium causing meningitis in a neonate, well, what I might do is I might make it my major case, then find other papers that say what other fungi cause meningitis in neonates. Or I might say what other things can scedosporium cause besides meningitis in neonates. I want some context to why that case is so interesting by finding other material. It won't always be possible, and there's more than one way to present the case, but it's often quite nice to make the case where your audience is working with you to work out what the answer is. So you may want to withhold the final cause. It may be on your first slide, but sometimes you might just say, strange case of meningitis. And you work through the case at the very end after you've got all your evidence together, all your tests to go, and the cause is, and that makes it for your audience more interesting. That's not necessarily the way you have to do it, but that's a way that can be quite useful to do it sometimes. When you do your PowerPoint, no more than 12 minutes. You'll be amazed, most students struggle to go 8 minutes. But no more than 12. At least twice in the last couple of years, I've had students rambling on for 20 minutes. And I had to say, stop, because we're all going to sleep. But 12 minutes, you know, as a maximum is reasonably good. That's quite a long time to talk. Be prepared to answer questions, and if no one else asks them, you can guarantee that I will ask plenty of questions. So make sure you know your material well, because I'll be marking you on how well you answer your questions. The one thing I won't mark you on is how well you present, because it's a skill it takes time to acquire, and sometimes you're just nervous or you're not very outgoing and you find talking really hard. You aren't marked on how well you talk, you're marked on the quality of your slides, your content and your layout, and you're marked on how much you know about your topic, but not on how you present. With the slides, keep the amount of text per slide to a real minimum. The worst thing is slides with just full of stuff that just go, it's just overwhelming. And whatever you do, and I know that some staff who do this don't read the slides. What? If you're a student watching someone give a presentation and all they're doing is reading the text that you can read at the same time, is that there you're heading? I just can't stand that. So don't read the slides. Make the slides be dot points that you might talk to that might prompt your memory, but don't read the slides. If you want to have cue cards to help you, that's fine. But even worse than reading the slide is reading your own cue cards, like an essay. Just talk, and it'll be more fun. Make the headings and the size of the text be roughly like those. It's not a guarantee that it's going to have to be that way, but too small a text is often a real no-no. Use plenty of images, plenty of diagrams, timelines for cases can be quite good. Say, they will sick on this day, this specimen on this day, this result on this day. Show me how it unfolds over time. So, okay. Okay, so as well as the PowerPoint slides that you'll present to us, you must prepare a 500-word text handout. This is basically an abstract of your talk that all the students will have in front of them while you're talking, that gives them some of the content in case they miss what you're saying. This must fit on a single A4 page. So even if you've got 500 words that goes over the page, make it in one page. This is for my benefit, because I make a little booklet at the end of the semester before we do the presentations of all put together. And nothing is more annoying for me than having one that spills over on the second page. I would go in there, edit it, change all the pagination, can't handle it. One page per student, and it's easy. For your references, put them on a second page. I want to see them when I mark your work, but when I hand out them to the students, I'll remove the reference page by just deleting the second page. Don't put them on the same page under your abstract. That's all straightforward. What you'll send to me for assessment will be your Word document, your abstract, your PowerPoint slides, and a PDF copy of the major article, your primary article. I want to be able to read your article and make sure what you're saying to me in the presentation is not made up. And make sure you label them so I know what they are by you putting your name there. There's a whole pile of great journals that have great cases, and there's just some of them, but there's a lot more than that. They're ones that I use quite a bit. So you'll be able to find some of those that are quite useful. If you're doing a master's or a grad dip, your assignments are a bit different. For you, you must do an outbreak for your presentation, not a single case. It must be an outbreak. And you must discuss in your outbreak what techniques we use to link the offers together to prove it was an outbreak and not just a symbol assembly of separate cases. So when you read the notes there on the assignment, it's quite clearly described how you do it differently. And for assessment, it's half assessed on your text abstract and half on your PowerPoint slides and knowledge of the case. All good? Everyone happy? Lucas, mate.
Do the cases have to be recent, or can they be like historical cases?
All right, as a rule of thumb, I'd say the case should be no more than 10 years old unless there's a really good reason. Like, an old case doesn't necessarily mean straight away, no marks, it's an old case. There's got to be a good reason for it, because we've moved on a lot and there's better techniques available now, but there may be one you've got that's from a while ago that's just so fascinating. But usually I'll say within 10 years would be a good rough rule of thumb. Yeah, okay. Everyone's good? Right, done that, done that. Okay, now for some content. I have no idea why I've got Dora Explorer on my slide. I think at the time I was in my Dora phase, but I'm not quite sure what it's here for. So I want to give you an idea of why mycology is different than bacteriology at a rough starting point. So first of all, the specimens we get are quite different. So for bacteriology, specimens are swabs, or they're liquid, they're biopsies perhaps, or they're aspirates, or they're urine, or feces. With mycology, they're normally a much drier specimen. So swabs are hardly ever used in mycology. It will either be a skin scraping, or a hair cutting, or a nail clipping, or a biopsy. But a tissue, solid tissue. It's hardly ever a swab. Swabs are very poor specimens for most kinds of mycology. Think in a way, probably because not many fungal diseases make pus. It's not liquid exudate that you get with a swab. It's normally more of a rash or an outgrowth or some sort of scaly lesion that's really requires to be scraped off as opposed to being swabbed off. The swab's hardly ever used. We do direct microscopy, but not a gram stain. So in bacteriology, swab comes in, microscopy direct on the specimen to gram stain. With mycology, it's not. It's normally, depending on the kind of specimen and what the disease is, it would be a lactophenol cotton blue preparation. Cotton blue is just a blue stain. It's not actually a good stain. There's no differentiation between different organisms. Just stains in blue makes it easy to see. And the lactophenol part of it makes the stain stable. Because with mycology, lots of times the specimen itself is quite thick and solid, and the fungal elements are inside the stuff, you can't stain them or see them until you do a digestion. So it's common with mycology, if the specimen is solid, to do a digestion in KOH. It's a strong alkaline solution that turns a piece of toenail from being hard and lumpy into a soft bit of mush. And how will you do a digestion for depends on the specimen. A piece of skin you might digest for a couple of hours. A big chunk of toenail might be overnight. And then you add your cotton blue, and then you put on the slide, squash it, and it flattens right out and becomes visible. The KOH will digest all the non-fungal elements, but won't digest the fungi. They stay intact. You can do other kinds of stains. There's one called Kappa-Fluor white. And Kappa-Fluor white is a non-specific fungal stain that's It's not immunofluorescence, it's just a fluorescent stain that binds to the cell wall of most fungi and not to other material of host origin. So it's not a bad stain because quite frequently with mycology you might have a serious infection but a terribly small amount of fungal elements within the specimen. So it's people are doing a TB Gill-Nielsen stain. Lots of pus there, small number of organisms, hard to see. This makes seeing your bits of fungi easier because you see a bright white fluorescent thing in a black background. But of course it requires you to have a fluorescent microscope. So it's not a bad technique, but not that widely used for that reason. If you're doing biopsies, then you do histostains. Silver stains or PAS stains are good at picking up fungal elements, but only in biopsies. When it comes time to culture them, if you're culturing for fungi, there's a very, very limited amount of primary culture media. The whole pile of media you use later on for subculturing, but as primary media for isolation, there's not many media. Most media is based on Sabouraud's agar, SABS agar for short. And SABS agar is selective in the sense that most bacteria grow poorly on it. It's too sweet. It's got lots of sucrose in the media. We can also make our SABS agar more selective by adding various antibiotics to it. So plain SABS is a little bit selective in the way that most fungi grow well and most non-fungi don't grow very well. You can make it better by including a broad spectrum antibacterial drug. Either chloramphenicol, it's getting hard to get hold of these days, it's becoming less and less commonly used, or gentamicin. And what those drugs do, they're broad spectrum and inhibit most bacteria, but have no effect on fungi at all. So in terms of mycology, a SABS core or a SABS gentamicin plate are non-selective because all fungi grow equally well. There's one particular kind of fungal group, and they're called dermatophytes. And dermatophytes cause most cases of tinea. Dermatophytes grow quite slowly compared to other fungi, which we call saprophytes, that are common contaminants. Aspergillus and penicillium are common contaminants. They're saprophytes. To make you be able to grow dermatophytes, pathogens, slow growers, in the presence of common contaminants, non-pathogens, and fast growers, you want some drug that selects for dermatophytes and negates saprophytes. And that drug is cyclohexamide. So you do have occasionally the requirement to use a SABS agar plate that contains cyclohexamide. And it's normal to have in that plate also some gentamicin or chloramphenicol to inhibit bacteria at the same time. And we call those plates mycobiotic plates. All that is SABS plus chloramphenicol or gentamicin plus cyclohexamide. And that plate then becomes selective for dermatophytes against saprophytes. And these plates are used whenever you suspect the person's got tinea caused by a dermatophyte. That'll become clear next week when we do dermatophytes to understand what tinea really is. So as well as having either SABS or SABS plus different drugs, the other media we use occasionally will be some kind of brain-heart infusion agar. Very occasionally the fungi we're growing are fastidious and need nice stuff to grow. SABS agar is pretty much not that nutritious. But most fungi don't care because most fungi are really good at just growing on anything. For the kinds of fungi that need more stuff, brain-heart infusion agar is a bit like the blood agar version in bacteriology. It's an all-purpose medium. Grows everything. And after you put your stuff on the plate, you incubate. For how long? Most fungi grow slowly, so even a very fast fungus, well, the ones that are super fast, will fill the entire plate and lift up the lid in 2 days, and they're uncommon. The ones that we see that cause tinea take anywhere from 3, 4 to 5 weeks to grow. And that means a colony the size of a 10-cent piece, 20-cent piece. They're slow. And some others take 6 weeks to grow. So growth is slow. Because growth is slow, there's 2 ways to incubate your plates. Well, there's 2 ways to incubate your media. One way is to use a plate. And in 6 weeks' time, the plates will dry out. So we normally seal the plate with sticky tape around the outside. For teaching, I use plates because plates are easy for you to see and look at them. The risk with plates is that when you lift up the lid, all the spores of the fungi come out and you breathe them in. For our practice, it doesn't matter because I know what the fungi are and they're safe. But in a real laboratory, there's always a risk that you might have a fungus that can cause disease in humans by inhalation. You don't want a plate of a fungus that you can inhale and cause disease. So most labs use not a plate, but an agar slope in a sealed tube. And an agar slope is just going to be a 20mm tube, screw cap lid, slope. This is better because it won't dry out and the surface area is low, so less aerosols generated, and you look at the whole thing without pulling the lid off because it's just safer. So labs will use the slopes, but in practice we'll use plates because it's better for teaching us all. And you normally incubate for at least a month before you discard them, again depending on what your suspicions are about what the disease might be and what the specimen is. Okay, so you've got this stuff, you've done the right thing. Most fungi incubate at room temperature. What's wrong with room temperature? It's a stupid concept really, isn't it? Because what's room temperature? Depends where you live. In Tasmania it might be 5 degrees or less. If you live in the tropics, it's warmer. So, good labs have an incubator at room temperature. And that's normally about 25 degrees Celsius. Between 20 and 25. And most labs have got air conditioning and central heating. Their ambient temperature will be in that range. So for us, all we do is leave them on the bench. There are a few fungi that need to grow at 37. And again, when you encounter those fungi, you normally know by the kind of specimen that you have and the kind of disease and presentation. But it may be that for some specimens, you do more than one plate. You do one at 37 and one at 25 or 20. The problem is, one of these fungi that grow at room temperature won't grow at 37. Now, most fungi in their normal habitat are in the environment. And on a human, they're growing out of their comfort zone. So even though you might have a dermatophyte, for example, that grows in your skin, just there is not 37 degrees Celsius. And these fungi that cause skin disease often won't grow at 37, even though they're growing in your body. But they're growing in the cool part of your body and your skin where it's exposed. All right, you've grown something and you're pretty happy about it. How do you know what it is? So what tests do we do to identify your organisms? First of all, mouldy tough is not good yet for fungi. It's okay for yeasts, but not for moulds. And most of our work is on moulds, long, furry fungi. I think as time goes on, it's going to get better for mouldy tough, but at the moment it's not great. So we still require other kinds of systems for identification. And they both really involve lots of observation. And if I say one thing about mycology compared to bacteriology, it's like this. With bacteria, the tests you do are positive or negative. And you might do a whole lot of tests and get a whole lot of pluses and minuses, or you might do them in sequence. This is positive, therefore do this test, that's negative, do this test. With mycology, the tests don't often give you a plus or minus answer. They give you an observation. Are the spores long or short? Oh, they're a bit short. Are the spores round or oval shaped? They're oval shaped. It requires lots of interpretation and observation. It's harder in that sense than bacteriology, and it can't be so easily automated. So once you've grown the colony, your first test is microscopy. Because most mycology relies on you seeing what kind of structures the fungus makes to reproduce. If the fungus is not reproducing, you can't know what it is. Because all identification is based on what kind of spores it makes. Does it make round ones or long ones? Big ones or small ones? How they formed on the structures, do they form the hyphae or they form the small branches? Is the hyphae curly or is it brown? Is it straight? It's all about observation. So, microscopy. So, the most common way of doing microscopy is called a tape prep. And for a tape prep, you have your plate, there's your fungus on there. You take some sticky tape, a small square of sticky tape, and you place that on your fungus, sticky side down. And you pick it off, and you put it on your slide, sticky side up. And hopefully you transfer all the hyphae and all the Candida from the colony, and they're now on your bit of tape in the same arrangement as they were on the fungal colony. Then you add some stain to that, and it covers it, and you have a look. It's all about observation. And for those stains, we use cotton blue or nothing, because with good phase contrast microscopy, the stain becomes less relevant. Sometimes on your primary plate, which might be a SABS agar, for example, or a microbiotic agar, you do this test and you see nothing. All you see is hyphae, because for whatever reason, the fungus decides not to reproduce on this primary media. So that means right now, you've got nothing. So we often subculture these fungi onto some more media. And there's a whole range of media that's been designed for various purposes, but primarily to make the fungus reproduce. So we may use a plate like honey and milk agar. It's called lactic milk. It's honey and milk, and it makes the fungus happier, and they reproduce. We often use tap water agar. And tap water agar is just tap water and agar. It makes them very unhappy, so they reproduce. And then you see some spores, and you know what's going on. So these are specialised media designed for a certain purpose. Very occasionally, there may be one or two special tests that you do that might be a key test in clinching your identification. But most times it's about this: microscopy, subculturing, and more microscopy. A very, very small component of identification is colonial morphology. So in bacteria, it's all about plate reading. Yet the colony does this, and does this, and does this. With mycology, it's less important because a lot of fungi are simply white and furry, or a bit brown and furry, or a bit red and furry, but it is furry. So the colonial morphology is helpful sometimes, but not such a big deal. Still record it, it's not hugely important. From the time you get your growth on your primary plate, if things go well for you, you might get an identification in 2 days from then. It might be a week, it might be 2 weeks. It tends to be slow. If any test that you do requires growth and the fungus is slow, add 2 weeks, get some growth. All right. So the fungi themselves are a whole separate not stain well with a gram stain. Now, if you do a gram stain on a specimen that contains fungi, you see things that are bit gram plus bit gram neg. They do stain, but the staining is inconsistent, not reliable. So we use up the stains. There are three broad kinds of fungi, in a really broad sense. Some are unicellular, and they're yeasts. Yeasts are easy, because yeasts behave like bacteria. They grow on plates, they're blobs, you can do subculture with a loop, and strip them out, you do API tests. In general, for most purposes, these are treated the same as we treat bacteria. You do plates, you do lawn plates, you can do diffusion tests, API tests, biochemical tests. They're relatively easy. And the shape's either going to be oval or round, but not much variation in them. The ones that for us are more challenging are the ones that are multicellular, and we call these moulds. And a mould is defined by having hyphae, and hyphae are long multicellular extensions of the organism. They're like branches of a tree. And they have septa. Some have septa, most have septa. And a single thing is called a hyphae. A bunch of hyphae together is called mycelium. So, that's hyphae, but that's mycelia, plural. And the entire colony that you see visibly is called a thallus, the whole thing. So we've got yeasts and we have moulds. There's also a very small subset of organisms that are dimorphic. There's two broad kinds of dimorphism. One is real and one's pretend. The real dimorphism is like this. We call dimorphic fungi thermally dimorphic. A fungus that's got thermodimorphism in the environment, its normal habitat, the organism exists as a mould. Out there in the ground, in the dust, in the trees, in the birds' feathers, it's a mould. When it affects humans, and is under the stress of a change of temperature, it will convert to a yeast. It's the same organism, but it can just grow in a different form depending on where it is in its cycle of life. So these are moulds in the environment. They're infectious as moulds. You breathe in the spores to get infected, but then when you're in your body, they convert from being a mould to being a yeast, because a yeast is better at growing inside a body away from open air spaces. I mean, fungi are aerobic. They want to grow in and carry with some air. If they're in a some sort of body, they convert to a yeast. A small number of fungi can occasionally appear to be dimorphic, but they're not really. These are pretend dimorphics. And these ones are ones that change morphology as they get older. So sometimes a mould, as you grow the plate and it gets really, really old, goes moist and becomes yeast-like. And sometimes a fungus that's a yeast, you look a long, long time, they go furry and become mould-like. They are not really dimorphic. They just appear to have different stages of growth depending on time. True dimorphics, and there's only 5 or 6 of them, they are dimorphic based on temperature and location of the body. Just wonder if now's a good time for a bit of a break. You're all a bit fidgety. How about we have a break for about 10 minutes now? All right? So come back at this clock at 1:15. Okay, let's move on. This slide has the potential to be confusing, but it needs to be. So the first thing is, this slide has almost no relevance at all to how you practice mycology in a laboratory. Okay? But it's important if you're doing reading to understand what this stuff means. But it doesn't really affect what you do in the lab. So if you don't get this at all, then ignore it, and you'll lose 5 marks in the final exam. It won't be a deal breaker. So the first issue is, with bacteria, they divide by fission. That is, 1 cell becomes 2 cells, becomes 4 cells, and every single cell is the same. Fungi can reproduce sexually or asexually. Depends on their mood. And sometimes the morphology and the appearance of the organism will change depending on whether it's reproduced sexually or asexually. As a consequence, some fungi have 2 names. One name for the sexual stage and one for the asexual stage. Virtually all the fungi we see in human disease are in the asexual stage. So for us, it's not really that relevant, except that by a weird quirk of how the whole science has been devised, if a fungus has 2 names, often the name of the sexual state has preference over the one that's asexual. So sometimes, even though you see one version, you name the other version. It does my head in, it's just a stupid system. But you'll often see one bug referred to by more than one name. That's all you have to understand. So what we see usually is the asexual stage, and what we normally name them as is what we see, the asexual stage. In terms of how we classify the fungi into broad groups as a taxonomy, the taxonomy is based on how they reproduce sexually, not asexually. And there are 5 broad kinds of fungus based on how they reproduce sexually. Zygomycetes, ascomycetes, basidiomycetes, mycophycetes, and gyronomycetes. This class of fungi, called the gyronomycetes, are also called the fungi imperfecti. This is because most of the fungi in this group are put there because they have no sexual state. They really they don't fit anywhere in those spots there or those spots, put them there. And this term means non-sexual. So the teleomorph, in other words, the sexual state, is also often called the perfect state. And the anamorph, the asexual state, is often called the imperfect state. Now, you'll be amazed how many people who are mycologists who spent their whole life trying to make these fungi have sex to prove it they can do it. And they've been successful. And you'll find now some of these fungi have a second name. And they forced them under all weird conditions to reproduce sexually. It's not got any basis in practical everyday mycology, but you'll have to understand what this means when you read stuff, if you ever do any reading. All the fungi in this group are the ones we call dermatophytes. So even though this is kind of a default collection of organisms, they are related in the sense that they are the ones we call dermatophytes that cause disease on our skin and cause tinea. When a fungus can reproduce sexually, there are 2 kinds for this. One is homophallic and one is heterophallic. A homophallic reproduction is when within one single colony the fungus will reproduce itself but still sexually. Other times it takes two different fungi to reproduce together, and we call it heterophallic. Again, it has no real basis in what we do in a practical sense. So that's a rough idea of how it works. Okay, this is a microscopy of a common fungus. And these names for you at the moment, don't worry about writing them down and get panicking about them. The names will go over again at the time it's more relevant. But we'll just have to go used to these names. This is called Scedosporium apiospermum. And this fungus in this slide, these spores are asexual. This is what you see for most patients. It's quite an important cause of disease, quite nasty, really hard to treat. And you see hyphae there, you see septa in the hyphae, and these long things here that give conidia. A conidia is a separate term for a spore. So as a science, mycology wants to become a bit sexier. Rather than saying spore, which everyone knows what it means, I say conidia. It means the same thing, really. So you'll hear me say spore or conidia, and they mean the same thing. This means it's a cell designed for reproduction. And you'll find here that some are formed at the tip and some are formed laterally. So I'll call it a terminal spore or a lateral spore. So for the next little while, all I want you to do is get an idea of the kinds of observations you're going to make to put fungi into certain groups. So this is Scedosporium apiospermum. If you're lucky, and I've tried this every year for 15 years and gave up a couple of years ago, you can make this fungus reproduce sexually. I can't, but you can if you're clever. And then you see this. This is the same fungus, but now it's reproducing sexually. And rather than forming these spores that are asexual, is forming these ones. And these are called ascospores, which is why this fungus is an ascomycete. And now the name is no longer Scedosporium apiospermum, it's now called Pseudoalacearia boydii. Love the names of the fungi, they're really good, they're cool. So this is the same organism, different name, different morphology. But you would not see this in a patient unless you were incredibly lucky, because most times it grows in the way that's asexual. So in terms of how we identify fungi, the first is it's based on morphology. And most importantly, it's the morphology on microscopy, how they reproduce, and it's almost always based on the anamorphic, asexual stage, not the telemorph sexual stage. It's based on what you see normally, which is asexual, the anamorph. And a propagule just means a cell designed for propagation. In other words, a conidia or a spore. Same thing again with mycology, lots of terminology. And I hate the terminology, so I use it sparingly. If you do some reading, you'll find so many terms that can confuse you completely. I'll try and be sparing what I use, only use what's necessary. So there are two broad kinds of asexual propagule. One is called a conidium, or plural is conidia, and the others are sporangiospores. They're a bit different. Conidia are not contained in a sac. They're just formed either in chains or clumps or clusters, but they're single. They're not single necessarily, but they're not enclosed in anything. A sporangiospore is a spore What you want to see is, does it make lots of them or not many? What shape are they? Are they round or oval or funny shaped and curly? Are they small, and we call them microspores or microconidia? Or are they big, and we call those macrospores or macroconidia? Or it may be that one fungus can produce two kinds of spores. It might produce microspores and macrospores. So, what are they? How many? What shape are they? Big or small? And then look at how they're produced. Are they formed in big sacs? Are they formed in chains or clumps? Are they formed on the hyphae or on the end of different structures altogether? It's all about observation and recording what you see. So it's how they're formed, how they're arranged, and their morphology, size and shape. With bacteria, we've got this idea, yep, there's four kinds. Gram-plies, gram-neg, cocci, bacilli. Well, in a similar way with mycology, the first point for me of observation is the kinds of hyphae. This is really useful. There's a big, broad division in what kind of mould it is, depending on its hyphae. Hyphae can either be hyaline, non-coloured, clear, or pigmented. One of the two. There or there. Two types. And it's either septate or relatively non-septate. Most fungi are septate. And the only ones that are non-septate are so different it's obvious. So because the septa in fungal hyphae can be hard to see, I decide whether they're septate or non-septate, not on the basis of seeing the septa, which would seem logical, on different morphology, different criteria. So it's like this. If the fungus is non-septate, it's clear because all the ones that are non-septate have very, very broad hyphae, huge, fat. If I see big fat hyphae, I go, yeah, it's one of those, it's non-septate. Even though sometimes you might see one septa, because non-septate means not many septa, very few. If the fungus hyphae is not like that, not big and broad, then even though I can't see septa necessarily, I call it septate, because by default, most fungi are like this. So you say to yourself, okay, is the hyphae this or this? My first question is the hyphae big and broad and fat like a ribbon? If the answer is yes, it's one of those. Answer is no, it's one of those. With the colour, again it's a bit annoying. There's colourless and there's a bit brown. The brownness can be hard to see, especially with a blue stone on top of it. So, if I'm not convinced that the hyphae is brown, I call it hyaline by default. And the fungi with brown hyphae tend to form black-brown colonies. Makes sense. The only confusion is there are some fungi that are hyaline hyphae that make black spores. And then the colony is black, and you might think that the black is the hyphae, it's not the spores. The spore colour is irrelevant to this grouping. That might all sound really vague, and I'm sorry, but that's kind of the way it is. It's important you understand what's vague, because if I tell you this stuff is really obvious, then some students, and in particular, will get in the back and go, but it's not brown, it's not brown, not brown, and go to meltdown. Sometimes the hyphae is brown won't appear brown, and it's okay to be confused, honestly it is. And then, much less important is the colony morphology. What's the texture like? And this can change. So, even with microscopy, how a fungus appears on microscopy can depend on so much. Things like what media is it grown on, because some fungi will form some spores on one media and not the other. And same with morphology. It might be a different texture in the old colony. So whenever you record anything as an observation, you must say how old the colony was and what the media was. Otherwise it has no meaning at all. And the idea that sometimes the fungus is cottony, in other words it's furry. It might be very, very fine like velvet, we call that velvety. It might be lumpy like sandpaper, we call that granular. It might be smooth like leather, we call that glabrous. These terms are not strictly defined terms. What I might call cotton, you might call velvety. It's not that critical, because one single fungus can have a whole variety of different kinds of morphologies depending on the media anyway. But they can be helpful sometimes. Same as the topography of the colony. Sometimes in cross-section it is flat. Other times they're quite heaped and folded and lumpy. So how it appears in relief can be important. Again, it's not super important. The major factor always will be the morphology of the conidia on microscopy. And the colour, you always record the colour on top and reverse, because quite often there'll be pigment produced but only visible on the underside of the colony, not on the top. So when you combine your observations about microscopy and the colony morphology, you sometimes, if you're lucky, get an answer as to what the fungus might be. But it's not easy. For us in the lab, what's more important is that we have the fungi grouped into how we might see them based on specimen type and disease type. So when we study all the fungi, we won't study them by their taxonomic groups, or study them by what diseases they cause. So sometimes the infection is what's called superficial. In this case, a fungus is growing on your body somewhere, and you're not happy because you look funny. But you aren't in any pain, there's no itch or lesions or anything like that. It won't kill you, but you hate this big rash on the end of your face, or you hate having these curly toenails. We call those things superficial. They tend not to elicit an immune response, but they can be cosmetically uncomfortable. So therefore you want them to go away. And then there's a whole section called cutaneous disease. And most types of true tinea are cutaneous disease. And this means the fungus is now growing not just on your body, but in the tissue. However, it's only growing on the very outside surface, the dead keratinized tissue. So the dead parts of your skin, or your hair, or your nails. So cutaneous disease involves skin, hair, or nails. And you do make an immune response. And if you leave them, they will get worse. They're not going to kill you though, but they do get worse. And then some are subcutaneous. And for things that are subcutaneous, normally it requires some kind of trauma to implant this fungus deep inside your tissue, something below the surface of the skin, in muscle, in bone, in the fat layers. And when it's subcutaneous, these can be horribly disfiguring. And if you leave them and don't treat them, they will spread and get worse, and they can be horribly nasty. And then there are some that are systemic. There's more than one kind of disease that's in that way systemic. The most common kind of systemic disease are the ones caused by the thermally dimorphic fungi. And with those infections, you breathe in the spores from the environment, and the bug undergoes a change of morphology into a yeast form, and then the primary disease is in your lung, and from the lung it can spread anywhere, often from your lung to the blood to the skin. So even though the infection was acquired by inhalation and the primary infection was respiratory, often your first thing that makes you feel uncomfortable is big lumpy wrinkles in your skin. But it's got there from the inside, it's come out that way, not from that way. And then increasingly now, there are so many fungi that in the past would never cause disease that now can cause disease, and these are totally opportunistic. We're getting so good at keeping sick people alive that fungi end up taking your ability decaying log and try and grow on you or in you. And these things are horrible because the people are sick already, and these fungi are really hard to diagnose, hard to treat, not nice. Some terminology. If you go to a website or any kind of book and you go to the glossary, there will be pages and pages of terminology. So much so that you'll just drown in it. This is a selection of things that we use a lot, so you want to know what these mean. So conidia, or singular conidium, just means a asexual propagule, so it's a cell that's formed asexually that then can give rise to a new organism. It's reproductive, but it's asexual. And by definition, these cannot be enclosed in any kind of structure or sac. It's a very broad term, conidia. It can be big, it can be small, all sorts of shapes. Just a very broad term. Sometimes, here's your bit of hyphae, there's some septa. It may be formed like that on the hyphae. That is a conidium. Other times, there'll be a separate structure, specialised, that forms conidia. And we call this thing here, this specialised branch, a conidiophore. In other words, it's a branch that forms conidia. And these might be chains, they might be clumps. Sometimes the chains that get formed, and you can never tell this from microscopy, but it's worth knowing this. Sometimes with these chains, the newest cell is here, which means they're formed inside there and popped out. As the chain grows, the newest one is just there. Other times they grow at the other end. The newest one is there. You can't tell by microscopy, but it is different. And there's a term for that as well. I don't know what it is. Sometimes a fungus can form a new cell by having the hyphae break off like that. And because they're formed by joints, we call these things arthroconidia. If you're a yeast and you form a second cell by budding, we call that bud a blastoconidium. There are sometimes in some fungi a funny thick-walled structure which was thought to be some kind of form of reproduction that's called a chlamydoconidium. As it turns out, these are storage granules for nutrients. They're not involved at all in reproduction, but this term is stuck. They can either be inside the hyphae, and we call them therefore intercalary, interhyphae, or they might be terminal at the end like that. And they're often quite thick-walled. The term hyphae just refers to the long extended multicellular filaments that form the fur of the fungus. When it's together and visible, we call that mycelia. The whole thing's the thallus. Sometimes you may see a yeast like this. and it forms a bud that won't break off. Stays at home. And then another one, another one, another one. This might look a bit like hyphae because these are just yeast that haven't broken off. True hyphae has no indentation at the point of septation. These buds that haven't broken off indent. So we call this pseudo-hyphae as opposed to true hyphae. And then you only find sporangiospores and sporangiophores in the kinds of fungi that have the big, broad hyphae that's non-septate. Big, broad hyphae, almost non-septate. Big, broad branch, great big sac full of spores. These spores are called sporangiospores, and the sac is called a sporangium, and the branch is called a sporangiophore. And this class of fungus is called, or they're called zygomycetes. And they're special because they always have sporangia and sporangiospores. They always have long hyphae that's non-septate, and they're fast growers. They're common saprophytes. Oh, before I forget, for the prac next Monday and next Tuesday, part of the prac involves you bringing something to culture. I want you to bring in something that's mouldy. It's mouldy food, or mouldy anything you like. Mouldy bread socks if you want, doesn't matter. But mouldy something to culture. Food is good because you find lots of nice fungi in food. Try and avoid mouldy bread because all you'll find in mouldy bread will be aspergillus. Mouldy oranges, all you'll find is penicillin, and there's Borman's bacteria. So bring in something that's like some cheese or some bananas, some strawberries, some tomatoes. I don't know whether it's going to be brown fur or white fur, but generally the ones that are green and furry are the ones that are least interesting. It's all you've got, it's all you've got. But bring some food in, we'll culture it. Over the years I've got my best fungi from pracs, which is brought stuff in. Some nice fungi. You often find these zygomycetes on strawberries. So in your fridge, a strawberry that's got really sort of a dark grey colour with great big lots of fur, it's one of those. And, you know, for human disease, all the things that are opportunistic that cause horrible diseases, these are fungi from the environment on food, for example. So it's worth seeing them. This is a man with tinea, and this is caused by a fungus. It's a dermatophyte. It would be itchy. When you scratch this, you know they've got spores on your fingernails. And you scratch there, put some more there. Some more there, and some more there, and some right there. Tinea will spread because as the growth occurs in your skin, it's making candida, which then can translate somewhere else, cause disease, either on your body, or on the sheets, or on the towel in the bathroom, and you get somebody else. So most kinds of fungal diseases are not contagious, but tinea is. Bless you. This is subcutaneous disease. So all these growths here are caused by fungi. And this would have occurred by some kind of trauma that's implanted these fungi into the tissue from walking on a stick or a splinter or some kind of traumatic event. From infection to this presentation might take 3, 4, 5 years. So quite often they're chronic long-term diseases. They don't occur overnight. And that is a systemic disease. This may have taken 10 years to get this bad. But this occurred because this person breathed in some spores from the environment, was unlucky, went to his lung, spread from there to the blood, then came out from the skin. This is a disease of a fungus by inhalation. And this one is one that's dimorphic, thermal dimorphism. And these are endemic in some parts of the world. The good thing is they're not endemic in Australia apart from one, which is pretty unusual. Here's a nice colony of a fungus. This is a fungus called Microsporum canis. It causes tinea after handling dogs and cats. And sometimes it's this morphology. So I would call this velvety. It's not very furry, it's lightly furry. And I call this rugose. It's folded and lumpy. It's going to be hard to handle because it's very hard to subculture this kind of fungus. It's all leathery. Get your little needle, try and get some more fur off this, and you've got to cut a big chunk of agar out to transplant this to somewhere else. Believe it or not, this and that are the same species. And this one has just got sick of being like it was before and has got older, and often with fungi when they get old they become sterile and they make sterile mycelia. When you see all this white fluff for us is always bad news, because white fluff means not many spores happening. I like coloured, dense mycelia. White fluff, no spores. I prefer the first one. I love this one. This is Microsporum gypsium. It's a fungus that's grown that you find in the soil, can cause tinea. And what you see now is it's granular. You can imagine in this plate and tapping it, and again, it's all, this stuff here is all spores. You love these. Word of caution though, the microscopy of this bit and that bit are quite different, because here it's quite fresh growth because it grows outwards. Not many spores there, lots there. So with microscopy, it's normal to do a cross section. Put your bit of tape and cover a broad part of the colony, so you've got more than one part of the age. Because what's there is different. And in the very middle there, it's really, really old. It's gone white and furry. So that bit there, just plain mycelia out there where the action is. That growth there is 2 months of growth. This is a fungus with brown hyphae. If the fungus has brown hyphae, we call that demathiaceous. D-E-M-A-T, demathiaceous. D-E-M-A-T, demathiaceous. It's not derm, it's D-E-M, demathiaceous. It means brown, in other words, like earth. And this hyphae would be brown. Here's one with a pigment, and most fungi are non-pigmented. If they are pigmented, the colony is pigmented, but it won't diffuse. A very small number have a pigment which can diffuse out in the agar. So this has gone red there, and the red is leaking out in the agar. It's pretty unusual. Here's the microscopy. This is a fungus with two kinds of conidia. There are microconidia and macroconidia. So small ones and big ones. The small ones tend to be elongated. So oval shaped, often a bit rounder. And the large ones are multicellular. And one end is not round and one end is round. So I'd say large multicellular conidia the terminal end is round and the proximal end is truncated, snapped off. And this surface is like sandpaper, it's rough. And those things would tell me that this fungus is most likely Microsporum gypsum. You wouldn't know that, but what I'm saying is those observations, this shape, number of cells, all those things help you give it a name. It's all about observation and being careful. This is a fungus called geotrichum, and this is forming arthrochonidia, so this hyphae that breaks off into small sections, like little bricks. Each one of those can give rise to a new organism. This is an arthrochonidium. This is lovely. This one is a fungus with very broad hyphae. This is non-septate hyphae. Therefore, it's in this big group of zygomycetes. And this, therefore, must be a sporangiophore. And this big sac is the sporangium. And those things in there are the sporangiophores. This is a fungus called Apophomyces. It's a common contaminant. This is also a zygomycete. Long, broad hyphae. This is a sporangiophore. There's a sporangia, but the sac is broken off, and all you see is what's inside the sac, the sporangiospores. This fungus is hyaline, it's not brown. It is septate, purely because it's not the big broad hyphae. Therefore, by default, I go, well, it's not broad, therefore it must be septate. And you'll find that there's these long structures that come off the hyphae and give rise to small oval-shaped conidia, either singly or in clusters. This is called acromonium, can cause toenail infections. This one is brown. Keep the hands, brown. Yeah. So this will be a fungus that's demariaceous. It's got brown hyphae. It's not hugely brown, it's a hint of brown. And this fungus is called cladosporium. And what you notice here is there's hyphae, there's septate. You see little chains of hyphae, sorry, little chains of conidia. So we're seeing chains of conidia, and they branch. One chain there, one chain there. And they snap off, and either end's a scar. Little dark scar, like a little shield. And those things tell me that's cladosporium. So as a collective of observations, brown hyphae, demariaceous, chains of conidia that branch, so branching chains, and the single spores that break off have a scar. Cladosporium. So those things you observe and record tell you what it is. The hard thing though is there's no simple table. You can't sort of start at point zero and go, I'll observe this, therefore it's here, observe that, therefore it's here. The charts aren't like that. You must start with some suspicions of what the bug might be based on what the colony is like, what the specimen was, what the disease was like, and then from there you work out what the bug is. It's not as simple as just doing following a little flow chart. And this we saw before, this is Scedosporium. So hyphae, septa, spores being born terminally or laterally, but single. No chains, no clumps, single. As soon as you say single oval-shaped conidia, being formed on these pretty long ctenophores, I go, oh, Scedosporium. And now we're seeing these spores have more than one cell. They're being formed only at the end, not laterally, but terminally in small clusters. And there's no symmetry. They can have a bend at one end. So you can't always put a line down them and go it's the same on either side. So symmetry can be important, or lack of symmetry can be important. These are called chlamydoconidia, the big long storage granules that are thick-walled. And there's one again. This time it's inside the hyphae. We call that an intercalary chlamydoconidia. Almost finished for now. And now we're seeing the macrospores. Sometimes they're unicellular, sometimes more than one cell. And with imagination, this is like a banana shape. So our curved spores, multicellular, banana shape, fusarium. So same thing, it's the spore description which gives you the hint as to what it might be. Commonly causes rotting bananas and can cause human disease as well. Now I'm seeing like this, hyphae, septate. If this wasn't blue, it would be brown. This fungus is Dematiaceus. And this structure here is a specialised branch of the hyphae. And see this little hole there? Because these conidia here are formed inside and popped out. They don't grow on the end like you see before. These pop out into these big clumps. And this specialised thing here is called a phyllide. And these are called phyloconidia. Phyllide means vase-shaped specialised conidiophore. So a phyllide is a certain subtype of conidiophore. It's a branch of the hyphae with a specialised function to make conidia. There you go, finished. Right. We finished for now, the first 2 hours. I know this is a long 3-hour block, but I can't change it. I've tried for years. Just got to bear with me and just go with the flow. Let's have a quarter-hour break, otherwise you just get sick of it. We are already, it's all right. Do that. Come back at 15:15 on the dot, and we'll cover the further content that we need for the track on Monday and Tuesday. So about 15:15. Well, no, not about. 15:15 exactly. Okay, let's restart. Because the mycology is such a broad area, I know that students find it really hard to get their head around where the fungi sit in terms of what's what. So this little scheme is one that I've devised that works for me. It might work for you, it might not. If it does, I'm happy. If it doesn't work for you, I'm not happy. But I can't be even for everybody. So what I try and do is look at the fungi in terms of the most basic divisions possible. So my first thought is, okay, this is a fungus. Is it a yeast? Then that's one separate group altogether. They're easy. It's not a yeast, it's a mould. Very occasionally, if it's a mould, it will be thermally dimorphic. And these are so uncommon and unusual that you only see those in very specialised specimens and contexts that you will know if it's going to be one of these. So most times, if you see a mould on a plate, it's this group here. It's not a yeast and not one of those, just a mould. Okay? And these moulds are thermally monomorphic. In other words, these moulds are not those ones, they're just moulds. So if I see a mould, my first division in my mind is this mould one of those weird ones with big, broad, ribbon-like hyphae with no septa, or not really septa? That means it's non-septate, it's a zygomycete, and these will have sporangia and sporangiospores. So if you've got a mould, it will be obvious to you if it's one of these, because these grow really quickly, big colonies, lots of fur in 48 hours. So recognising one of these is easy. So the default normal process is, oh, it's a mould, it's one of these, it's not one of those, therefore it's one of these. So most fungi by default end up being in this group. And they're septate purely because they're not non-septate. Now your choice is like this. Is this fungus brown or black? Is the hyphae brown or black? If it is, then the fungus is called dematiaceous. And the other term, slightly more fancy for dematiaceous, is we call these phaeohyphomycetes. Phao means brown. And once you're in this group, then the number of bugs in this group and what names they are is a very, very small set. From down here, life becomes pretty easy. That's hard to work out, but at least you know what kind of fungus you're in. If you're not in there, then you're here. So normally the default is, oh yeah, it's one of these, it's not one of those, therefore it's one of these, it's not brown, therefore it's one of these. And these are the ones that have hyaline hyphae. These fungi that have hyaline hyphae tend to be shades of white, yellow, red, sometimes a bit brown, but brown from the spores, not from the hyphae. These can be pigmented, but the hyphae is always hyaline. And then you have one more choice to make. If this fungus is a dermatophyte, then the set of bugs you have is quite limited, because dermatophytes cause tinea, specialised disease. They always grow on plates that contain cyclohexamide, because by definition, dermatophytes are always cyclohexamide resistant. That's how you define them, basically. And they grow slowly. If it's not a dermatophyte, it's one of these. And the trouble is, this group here is massive. So most times you see a fungus, you can get yourself pretty comfortable. Oh yeah, it's one of those, small set of fungi. It's one of those, small set of fungi. It's one of those, small set of fungi, or it's one of those, I'm stuffed, because it's a massive group. The fungi in this group tend to be saprophytic. That is, they exist in the environment and break things down. They recycle material in the environment. And when they cause disease, it's normally opportunistic. That is, you're already sick for something else, and these just happen to come along and infect you because you're sick. So I have that map in my mind all the time. And if I was you, I'd print this out and have it in with you every prac and go, this fungus today, where is it on this chart? And I'll always tell you where it is. It will help you put things in little groups, and that makes your life easier. But when we look at fungi in the pracs, we look at them from the perspective of the patient. The first group today we're going to cover are those ones that cause disease that's superficial. And on top of that, we have these other ones we'll do next week, following week, following week, following week. So today it's this group here. Somewhere in between being superficial and being cutaneous is onychomycosis. Onychomycosis really means infection of the nails, either toenails or fingernails. And this can either be a bit cutaneous or a bit superficial, depends on the organism. So they sit in between, basically. All the things that are acute, superficial, the three ones we'll cover today: tinea versicolor, quite common, and tinea nigra and black and white pedra, very uncommon. So of those three, that one, common. These, much, much less common. That one there, exam, yeah, probably. These ones here, unlikely. So, again, remember, we'll get a specimen. It will be a skin scraping or a nail clipping or something like that. We'll do microscopy on it directly. And because of the risk that quite often, see, if I can make a cut right now, leave it on the bench for half an hour, incubate it, it'll go fungi, it's in the air everywhere. So, how do you know what you've grown on the plate is in your specimen or from the air? Often you don't know. And the only way of being sure is if it's airborne, it won't be there on direct microscopy. So seeing stuff on this microscopy directly from the specimen helps you prove that what you grow really is from the specimen and not from the air or contamination. Right, so tinea versicolor is a disease. The old name for the disease was pityriasis versicolor. And pityriasis is a term that describes fine, scaly skin lesions because for a while this was thought to be non-infectious. So pityriasis really is a dermatological term that says scaly skin lesions, but often not infectious because you can't grow this fungus very well, so often it was thought to be not infectious. As it turns out, this is a disease caused by infection from a yeast, and the yeast is called Malassezia furfur. And furfur is a term that comes from furfuraceous, which means fine scaly rash. I can't help it, these terms are in the books. But, so a furfuraceous rash means fine scaly rash. This fungus has had lots of different names. So older books will call it other names like Pityrosporium urbiculare, but the current name is Malassezia furfur. The other annoying thing about mycology is it's undergoing renaming all the time. And often one fungus will have 4 or 5 different names. One might be a new name and an old name, sexual name, asexual name. It does your head in, but get used to it. They have more than one name. And what you see in a person are skin lesions that can either be hyper or hypopigmented, depending on your skin colour. If you're a dark person, the lesions are light coloured. If you're a light coloured person, the lesions are dark coloured. And it appears to be that the fungus somehow interferes with the way your cells make melanin, therefore will change your skin appearance. They don't really itch very much, not itchy, they look funny. And it's common to carry this bug on your skin. It's normal skin flora. Something happens between you and the bug, and it stops being a nice, cosy arrangement, and it starts growing in a way that wouldn't normally grow. It undergoes some kind of transformation, and it must be linked somehow to your immune response, because we see these things a lot more in patients with immune disorders. It's also more common in summer than winter. 2 reasons this might occur. Maybe the sunlight is involved in triggering the transformation from being a normal flora to being a pseudopathogen. Or it may be that in summer, you get your gear off and you see the lizards that you don't see in winter. You've got clothes on. Don't know what the answer is. And there's a person who's got the disease. The trouble is there are other skin diseases that look the same, but that are not caused by Malassezia. They're just a skin disorder. And there's one called pityriasis rosea. We often get one lesion on your chest and it spreads somewhere else, and it's possibly viral, but it's not fungal. I remember I had it a couple of times now, but I had this infection on this skin condition on my chest, and I was convinced I had this. And I got the scalpel blade down, I scraped it away, couldn't grow anything, couldn't find anything. I scraped so much I almost bled from cutting my chest open. In the end, I realised it was not infectious. So the problem is, you want the lab to get the answer because how you treat this with fungal cream and the other ones with steroids depends on whether it's fungal or non-infectious. So you want to have the lab be involved in diagnosis to get the right kind of treatment. So these are lesions here which are hyperpigmented. Or if you're dark coloured skin, they're hypopigmented. They normally occur on the chest or on the back, less commonly on the arms or the face. And to diagnose them, it's relatively easy. You take a skin scraping, put the skin on the glass slide, add some KOH, wait for an hour, add some cotton blue, cover it, have a look. And what you see are lots of large round yeasts and angular hyphae, often called spaghetti and meatballs morphology. It looks like that, little bits of hyphae and spaghetti and these round little meatballs. A Woods lamp, a UV light source, will make your skin appear yellowish green where the lesions are. Can be helpful. If you want to culture it, you can, but it's very hard to do. The bug needs to have lots of oleic acid. So you do a culture by overlaying your plate with olive oil. But why would you bother? Because it's normal flora anyway. So growing it doesn't tell you anything at all. You must see it on microscopy. Seeing that disease, tilia versicolor, occurs in anybody without any immune disorder at all. But we do see it causing different kinds of diseases depending on whether or not you're immunocompromised. So we often have pilosporal folliculitis. So this yeast can infect the hair follicles, especially on your chest. And you get little pimples where your chest is hairy. You've got a hairy chest. Or you may get seborrheic dermatitis. So what this really is a skin condition where the oily parts of your skin start flaking off and it gets really uncomfortable and very itchy. This is commonly what we see as dandruff. So dandruff really is caused by malassezia. So when you comb your hair when you're a young kid, you used to have dandruff, you comb your hair and the white tufts come out. Those skin scars there are full of malassezia. And when you buy shampoo to treat dandruff, look at the name of the active ingredient. It's always called an azole, because an azole is a fungal drug. So dandruff is a fungal disease. So these are microscopy, large round yeasts, small bits of hyphae, often angular shape. If you see that, it's diagnostic, straightforward. There's a much more severe case. This person is probably immunocompromised. All these follicles here are infected. This is caused by the yeast. And there's basically dandruff below the hairline. It's seborrheic dermatitis. All flaky, you scrape that off, full of yeast. Somebody unlucky with it on their face. So this is tilia versicolor on the face. When I see that, what do you want to do when you see that photograph? You want to squeeze those blackheads and get rid of them, don't you? You just can't. I just go, oh, I want to get in there and get it. So that's tilia versicolor. Second disease is tinea nigra. And tinea nigra is a disease most commonly on the palms of your hands or the soles of your feet. So palmar for your hands, plantar for your feet. What you get are these lesions like you stain your hands with a bit of silver nitrate, just little brown blotches on your skin. And the yeast, the old name for the yeast was Exophialia wernickii. It's a fungus of stomatiaceous. The new name is Phaeoanlomyces. Pheo means brown. This is a black yeast And when you culture them, as it gets older, it gets more and more furry. It's not dimorphic, it's just the yeast that gets furry. And a furry yeast is because the yeast is forming pseudohyphae. If you leave it, it won't spread, it won't get any worse, it'll go away eventually. It's purely cosmetic. For the microscopy, if you scrape the lesions, you're going to see small little brown yeasts, often with a septa, so it's a double yeast almost. If you culture them, it'll be growing initially as a pale brownish sort of yeast. The older it gets, it might get a bit furry and become more hyphal. Fairly uncommon. There's the palms. That's me. You think, oh, I've just stained my hand with something. That's fungal. And there's microscopy, and you're seeing this is quite clearly brown. This is dematiaceous. Sometimes they're one single cell, others a double cell, but they're yeasts. There's the colony, where it's initially it's kind of globby like a true yeast. Gets older, becomes more furry. And there's microscopy from the colony. Always bear in mind, microscopy from the colony and the patient will often be very, very different. Often a massive difference in morphology. And then there's two kinds of piedra. Piedra means stone. So white and black piedra refer to a disease where you have nodules of fungal growth on your hair. And you feel it, so a little lump of growth. Because this one is a white little growth, it's often confused as being head lice. And they think that the little lump on your hair is the net or the egg of the head lice, when really it's fungus. So you'll see these little yeast growths, beard, scalp, groin, axilla, most commonly on the beard or the scalp. And when you squash this, so you take hold of a lesion, take hold of the hair, find the little white lump there, pick it off onto a slide and squash it. When you do a squash prep, you have lots of hyphae, and this proctosporin always forms arthroconidia. So it's quite straightforward to see it. If you culture it, lovely white yeast that goes furry. As an organism, this can also cause widespread full-body disease, but only if you're severely immunocompromised. So, really a separate issue altogether. There it is there. So if you saw that go, you've got nicks. But when you take one of those and have a look, it's not an egg with a lice inside, it's lots of hyphae. And if you squash that, or you grow that, there's the furry yeast. It's reasonably straightforward. Black piedra is caused by a black organism. And this time, the fungus is called Piedra horte. It's actually a brown fungus, and this is one of those rare occasions when the fungus is growing in the sexual state. And it makes ascospores, so it's an ascomycete. These spores are sexual spores. They're not asexual. And again, when you grow them, they tend to revert back to being asexual, and you tend not to see ascospores from a colony, but you do from the patient. There's a little nodule on someone's skin. Same thing, you take it off, you do a squash prep and you culture it. So you do two things, microscopy and culture. Now, so of those first three, tinea versicolor, pretty important. The rest, you've heard them once, forget about it. Onychomycosis now, this is important, it's very common. Onychomycosis is skin or fingernail or toenail infection. And because they're on your nails, they can be quite disfiguring. If your toenail's infected and goes all brown and curly, you don't care because you wear shoes. fingernails you care a lot because it's unsightly and you can see your hands all the time. There'll be a whole range of fungi that can cause nail disease. The two most common causes of nail infection are Candida albicans and the fungi we call dermatophytes. Now I'll go on and on about dermatophytes. Next week is all dermatophytes, you know what I mean? But for now, understand dermatophytes is a separate fungal group. And if you go back to my chart, oh jeez, a long way back there. Oh. They're here. So it's either dermatophytes, or it's Candida albicans, or else it's other. So the others involve a whole range of fungi. Scopulariopsis, Acremonium, Aspergillus, Fusarium. These are all hyaline hyphomycetes. Because your nails, toes especially, are dirty sites that contain lots of other stuff, fungi and bacteria, it helps to have your plates being selective. One problem is, all these fungi in this other group will not grow in the presence of cyclohexamide. So you must have a plate that's selective so you can grow your dermatophytes and not have them overgrown by contamination, but you must have a plate that's non-selective to grow the ones that are not dermatophytes. So if you only have your plate being selective, you'll miss all of these. I call that tunnel vision and cyclohexamide, getting rid of those other ones. The other problem is, if you do grow some of these can cause disease, but they're also very common contaminants. How do you know whether an aspergillus on your plate is really causing nail disease, or was there from the air, or there from their socks? You don't know. So the way to make sure you only see things that are really there is you want to see them in more than one specimen. And it helps to see them on the direct microscopy to support the growth being significant. Otherwise it might be contamination. So always do direct microscopy. And it may take overnight to digest a bit of nail. Look for things inside the nail, bits of hyphae. So if this was a nail that comes from a patient, what you want to do is the best specimen to get a not very sharp probe, a big chunk of, like a big thick needle with a blunt end, and dig in there. You want that muck under there. And you want to clip this nail off and get this. That nail there is still healthy. You want this one here and under there, and go for it, scrape it out. Because you're scared of hurting somebody, I just don't like it. If you're scared, say to the patient, do it yourself. Because they're more inclined to go right in and dig the bugger, whereas you're scared of hurting them. It's fine for them to do it themselves. Get right in there, dig it all out. You want as much as you can from inside there. You tend to find that the freshest fungus is here. Up there, it's getting quite old. And often it will be paused by microscopy. You'll see the elements there, but they're dead. It won't grow anything. So you want maximum specimen always. That's Candida. Horrible, horrible tinea. And those nails would be so crunchy. Flick those off, scrape them, clip them, but gather as much as you can. How gross is that? Sometimes onychomycosis spreads and involves the skin as well. And this is most likely a dermatophyte because most of the other fungi can do nail disease, but not skin. If it's doing nail and skin, think dermatophyte. And you want to scrape this off, trim the nails, get them off, and culture them. You know, suck those toes. Breakfast. Two of the reasonably common other causes of onychomycosis are monomorphic moles, septate, in that other group. You tend to find that Scopiopsis will cause the nail to go brown. So a brown nail is perhaps a white nail with acromonium. And inside the tissue, when you do a microscopy on a bit of nail, you'll often see conidia inside the nail. Whereas here you see these long, sort of fingery bits of hyphae. When you culture them, these are typically brown colonies that are powdery, lots of conidia, and these are more felt like, less furry, and they're white. And their microscopy is quite different. So we've grown something, we've got suspicion based on the morphology of the colony, now we do some microscopy. This is Scopiopsis. So the hyphae is not brown, it's highlight. It's not broad, so it's septate. So now on my chart, I'm down a little there. And I see, look, a little sort of funny shaped thing here. This is a conidiaphore, and they're conidia. And when they break off, they can be quite round, but I see at one end, they're lemon shaped. As soon as you say toenail, and you say chains of round conidia, some being lemon shaped, I go, Scopiopsis. So that observation of the shape of the conidia is important. So they're chains, non-branching chains. So when you see that, you go, there's some stuff there. You drill down deeper, you go, what am I seeing? I'm seeing chains of conidia. They're non-branching. They're round, but some are lemon-shaped. And sometimes they can be spiky on the outside. I can't see here, but they can be spiky. That is different altogether. That's penicillium, contaminant. The chains are much, much longer than you saw before. So that and that are different, but similar. This is some growth. This is growth. If I see any kind of pink colony, pink to me is pink and the fungus is highlight. Pink colonies, I think, fusarium. So I've grown this from someone's toenail and this is a microscopy. I see banana-shaped macroconidia. Sometimes there's more than one cell, but they're banana-shaped. Classic fusarium. If you're lucky and you grow it on different media, that's the best form of fusarium. And now they're clearly banana-shaped. From that to that is the same fungus, different media. That's on Sabs agar, that's on soil agar. And my favourite media for making fungus do the right thing is called soil agar. And all you do is go outside the lab at Micro there, in the garden, get a whole handful of soil, watertight it, filter it, use the water for the agar. Works beautifully. Makes the fungus form spores. Well, I say I don't do it personally, but it's done for me. This is acromonium. So what I'm seeing now is long, thin conidiaphores. Clusters of elongated, almost pear-shaped microconidia. Against all the observation. And right now your head's probably spinning, so it's all too hard. It's not as hard as you think. As long as you just go in a slow fashion and be methodical. And the last one, just to talk about very briefly, is a disease called Scotilydium. There are two kinds of this organism. Most likely, they're the same thing, but one is a non-pigmented mutant of the other. One is called Dimidiatin, and it's black, and one's called Pyomen, which is white, clearly. And this is probably a mutant with no pigment of that one. There's a whole range of other names you'll see in the literature in books. These are old names for the same thing. They're superseded. And this This fungus can cause a whole range of diseases. It's not very common. It may cause a similar thing to the palm of the hand disease, or it may cause onychomycosis. But when you grow them, they're very fast growing. If it's dimidiatum, they'll be black and furry. If it's the one that's non-pigmented, they'll be white, but tend to get grey as they get older. But they fill the plate very quickly. And when you do some microscopy on the culture, you see all these arthrochonidia. So you'll be seeing like this. So there's the lesions, either on the palms of the hands or the nails. That growth there, maybe 3 days, 2 days. It's fast. And you'll see lots of normally pigmented bits of hyphae that break off. So in the prac next week, we'll cover the fungi we've covered today that cause things like superficial diseases. So the first prac will be, first of all, a few techniques. Seeing if your fungi, being familiar with what's septate, non-septate, what various spores are like. Look, this is more information. I don't want to make this more hard than it's going to be. The old names don't count anymore. The old names. Nope. The trouble is, when you read books, you'll read both names. Or often a book will have one name and then brackets the old name. The book, you know what it is.